The Arctic is an ever-changing place. As a fisheries oceanographer, I'm currently interested in one particular part of the Arctic, glacial Greenland fjords. These fjords are critical and productive ecosystems that depend on nearby glaciers. There is already a historical record of these glaciers retreating very fast. In the context of climate change, there is a huge need to understand how their disappearance may affect the fjords and their ecosystems. Our National Science Foundation funded project, which is part of the NSF program Navigating the New Arctic, aims to do just that. The goal of our project is to study Greenland fjord system. We have targeted two fjords, one in the east coast and one on the northwest coast, which are dominated by large marine-terminating glaciers. We have a fantastic interdisciplinary team. There are physical oceanographers, glaciologists, atmospheric scientists, biogeochemists, and climate scientists. We also have an anthropologist and collaborators from the Greenland Institute of Natural Resources. Then there is our group here at OSU, the biological oceanographer. So we are the zooplankton and fish people. We have a total of 4 research cruises. 2 of those research cruises in 2023 and 2025 are in Sermilik Fjords, and 2 of the other research cruises in 2024 and 2026 are in Melville Bay. To prepare for the cruises, we meet monthly with all the project participants. We discuss the sampling site, the place where we are going to have the monitoring, and also how much sampling we actually have time to do. On our first cruise, we did 82 CDDs. When we were inside the fjord, the CDD went down to at least 400 or 500 meters. CDD collect water samples measuring salinity, temperature, and depth.
In our first field expedition, we towed about 36 bongo nets. So bongo nets are these 2 big nets that sit next to each other, kind of like this. We take them and we trawl them through the water.
It is essentially a fine mesh net that will retain things that go more or less from less than a millimeter in size all the way to maybe a couple of centimeters or so. For bongo samples, we pull out all the fish larvae and we try to ID them when we can. Sometimes it is hard to ID them, and so we have to do that back at the lab. We measure the size and then we preserve them in ethanol. The rest of the bongo samples goes into formalin preservative, and then it is brought back here to Corvallis. For the midwater trawl, we immediately on the ship try to sort all the catch, or at least a subsample of the catch.
We brought all of these samples back to Hatfields, where I sorted through all of them by major taxa. We found euphalcids, which are krill, copepods, chaetonaphs, amphipods, and plenty of other zooplankton. Now that we're back here in Corvallis, we took a subsample and we extrapolated from there to figure out how many of each taxa are most likely to be found in a sample. We took some of the organisms and brought them under the microscope, measured them to see how long they are, which can help determine maturity level, and then we went through each of them to try to identify them to the lowest possible taxonomic level.
It requires taxonomic guides for those particular group of organisms. It is a process that may take months.
We're looking at stable isotopes as well. Stable isotopes are variants of an atom with the same number of protons but a different number of neutrons in its nucleus. We measure stable isotopes of carbon and nitrogen in order to determine possible sources and different trophic levels. With stable isotopes, it's very much like you are what you eat. We're able to look at stable isotopes with a lot of the water samples that we took in the field. We filter the samples for phytoplankton because theoretically they are at the base of the food web. That's what the zooplankton eat, the fish eat the zooplankton, and you go further on up the food chain to larger organisms like whales and seabirds.
It is really important because one of the other things that we don't know much about this ecosystem is the degree of connectivity. Can these unique fjords with this marine-terminating glacier seed the shelf for having higher productivity, which we can then track all the way to higher trophic levels? What was a real surprise, big surprise, was the abundance that we were seeing out there at the shelf edge. So the edge here between the yellow and the blue color, where there is an abrupt drop-off in bathymetry, we saw these amazing abundances of zooplankton out on this shelf edge. And so there is clearly a convergence of these different water masses.
Our work is to define each of the water masses, and we're going to look at the zooplankton communities within each water mass to see how they change throughout the fjord. The water masses in the fjord kind of act like a layer cake with three major layers. As you get closer to the glacier, there's a very cold, fresh lens. Well, basically just meltwater coming straight from the ice and the glaciers melting there. And then you'll have a middle layer, which is from the East Greenland current, and that comes down from the North Pole. Brings a little bit saltier, cold polar water, so it's a bit denser than the meltwater above it. And then below that, there's a North Atlantic current, which brings in kind of like warm, relative water, and so that's usually at the bottom. And these different water masses create different ecosystems, which can change quite a bit throughout the fjord system. So it was pretty cool because you're able to see that as we collected the data in the field as well.
I remember as a child, I have this vivid memory of being on these fishing boats. I would look at the nets that they were pulling up and try to guess what would come out of these nets. And I would also try to picture in my mind what type of habitat these fish that were tangled in the net were experiencing when they were down at depth. Sometimes we would catch tons of fish, sometimes nothing. Why was that? What was the fish habitat like at depth? And as a child I was fascinated by these questions. And that's really what gets me excited about this work today. This idea of trying to understand why. Answering these questions will contribute to our understanding of Greenland's fjords and the ever-changing Arctic.